So I'd like to introduce our speakers now. We'll be moving on quickly. Atul Butte is the director of the Institute of Computational Health Sciences at UCSF. He's also Priscilla Chan and Mark Zuckerberg's distinguished professor. He's authored over 200 research publications and is the founder of 3 investor-backed data-driven companies and was elected to the National Academy of Medicine in 2015. He studied computer science and graduated with his MD at Brown University, and he went on to receive his PhD from Harvard Medical School and MIT. And I think you'll all also like to know that his parents are in the audience tonight. I got to meet them earlier, so welcome. And thank you for what you've done for all of us as well. Our moderator tonight is Elizabeth Lopatto, although her mother calls her Liz, so that's what I'm going to do. Elizabeth is the science editor of Verge, and she's previously written for Bloomberg News and Seed Magazine, and so please join me in welcoming Elizabeth and Atul Butte to the stage. Thanks so much. Please welcome.
All right. Good evening. So Atul, I'd like you to start with maybe the 3-minute version of how you came to be on this stage tonight. What is your 3-minute biography?
Yeah, so everything always looks back great, in hindsight.
Right.
But basically, I'm a computer scientist and I'm also a medical doctor, and specifically in genomics and precision medicine, this new field. But I started in 8th grade learning how to code. My parents somehow afforded an Apple II Plus. I never looked back, and there's some in the exhibits here. We moved up to a Mac. I did computer science as an undergrad. I was working for some of the tech companies, Apple and Microsoft, and then went on to medical school. And in the middle of all that, the Genome project started. Got very excited about learning the ultimate source code of humanity, right? That's what's in that DNA. And finished medical school, trained as a pediatrician, watching kids grow and not growing, fixing those things. And then spent 10 years at Stanford as a professor, and now I've moved up to UCSF about 3¼ years ago.
All right.
That's a quick bio.
That was beautiful. I want to talk a little bit about the source code of humanity, which is to say DNA. And I think that there's something that's been in the news recently that might help us do it a little bit. It is, of course, the Golden Gate Killer, who, if you're not familiar with, was responsible for at least 12 deaths in the 1970s and 80s, among other sort of mayhem. And he was, of course, recently identified using basically DNA. It was an open source DNA site. So one of the things that I just wanted to start with is, the use of DNA in forensic work is about as old as I am. This is not necessarily a totally new subject, but there have been these sort of big historical moments where it shifted in our society. What do you think those have been? Yeah,
okay, so first of all, let's just get level setting for the audience. What is DNA, right? So DNA is like, I think of it as recipes. It's a book of recipes. And so the book I usually use as an analogy is the joy of cooking, right? Most people have the joy of cooking. It's a more neutral book to mention than the Bible or the Quran. Everyone knows the joy of cooking. It's a definition of wishful thinking. We all have it in the kitchen. We never even open it.
I look at it lovingly.
DNA is like that too, wishful thinking,
yeah.
Right? So, most of the DNA, there are some recipes there, and the rest are the binding, just like the book, right? Table of contents and stuff like that. But those recipes, your cells use those to cook up the things that the cells need to make up. So about 20,000 recipes, they can get misspelled. And they're different. I wouldn't really say misspelling. It's like the word color. We spell it one way here. In the UK, they spell it a different way. Neither is wrong, but they're different spellings, right? And so it's about 4,000 copies of The Joy of Cooking. If you just count up all the letters, about 4,000. That's about 3 Statues of Liberties if you just stack up the book in every cell. That's your DNA. So the DNA by itself is interesting, right? And now, what were the magic moments? One magic moment was 2003. The Genome project ended. It took $3 billion and 13 years to finish. And with that started a race to how cheap can you get your genome sequenced by many companies around here and certainly up and down the state of California. We're down to about $499, right? Down from $3 billion. Okay?
It's not bad.
It's not bad at all, but it beats any Moore's Law curve. And of course, we have to pay homage to Moore here in this building, but this is way faster than any Moore's Law curve, right? When those things happen, people start to use the technology. But the key point, especially with the Golden Gate Killer here too, is that the DNA by itself is interesting, but you got to compare it to things, right? You see the spelling, but you don't know, well, what's right, what's wrong, what does this spelling mean, how do you pronounce this word? You got to compare it to other databases. That's why we have these databases.
Basically, you need context, is what you're saying.
Absolutely. The DNA by itself is tough to figure out what does it mean. You got to look at a lot of other people who have DNA and then start to make these comparisons, whether it's for ancestry, for disease prediction, all of those things need others in databases.
Yeah, so the technique that the police used to arrest the suspected Golden Gate Killer, and I'm saying suspected because obviously he hasn't...
Alleged, I guess, right?
Well, they've arrested people before and it didn't pan out, so who knows? But they essentially compared the DNA that they had from his crime scenes to DNA that was on a publicly available database.
That's right.
And I guess what I'm wondering is, all of this is very, very new. The idea of having your own DNA sequenced is very new. What does that mean? How should we be thinking about this? How should we be thinking about, not just the databases, but also the techniques that are being used?
So I think a lot of us are now in this world where we have a lot of measurements on ourselves, so we sense that we can learn more about ourselves. And we are using companies to help us get there, right? Companies, organizations, or healthcare systems, or nonprofits. And so the database in this particular example were individuals that were getting value out of this setup, right? So they were learning about their own family members, ancestry. We have many of these kinds of companies, nonprofits and for-profits. And those databases are there, and people give out their names. In this particular database, people want names out there so that others can find them if they're distant relatives and all the rest. And so here in this case, and the terms of service, again, what do you say on the internet? I'm not a lawyer, right? So I'm not a lawyer. But the terms of, my understanding is the terms of service for this particular website, was that you're only allowed to send in the data if it was your data or you had the rights to that DNA, right? And so in this particular case, I can imagine custody rights for a sample, a specimen, I would imagine could fit in there. They uploaded and they found a relative. And that gave them suspicion, right? And I think they probably had a set of candidates or suspects already, and this really narrowed things down a lot. My understanding is they got more samples and validated using conventional methods, but at least give them a clue where they never had a clue. And even since then, I think there have been a couple more of these kinds of cases out there.
That's right. We recently wrote, actually, an article about there was some fetal tissue that was found in a sewage system that they're now using DNA sequencing to look for the mother.
Yeah.
So
it is tricky, right? Because you're depending on these databases yourself if you use this website, right? You can upload your data. If no one else is there, you don't find other family members. You don't see your ancestry. You're depending on others having already done this, but then you're also giving up some kind of ability to block others from using yours, because you're using, you're also giving in some way.
That's right. There's like the interplay.
That's right. Now, those websites don't know everything about you, right? They only know what you've given them. They don't know your medical history. I mean, those are all things you choose to enter to get more value out of those systems as well. And so at some point, you put in what you get out.
Yeah. Well, I mean, here's the question. Maybe the easier question, maybe the harder question. But how should we be thinking about this ethically?
Yeah.
About these databases, about their existence, about whether or not we want to participate, and what that might mean for our own family members.
Yeah. So I think their database is going to be there in every sense, and we know all the stories now, right? It's not just DNA, but we're focused on DNA right now. I think in the end, this is a new world where we're thinking about this. Data has always been there. Companies, credit card companies, a lot of groups have known a lot about us for a long time. And once in a while, they surprise us with things. And then they're surprised that we're surprised, right? Because we should have read terms and conditions of service and all the rest. Who does, right? There's people chuckling here, so we know that. I think, so the ethics have, especially on DNA, has been written about for literally half a century or more. I think we're in a world where we sense we're going to get some value out of participating in these kinds of constructs, whether it's a for-profit like an Ancestry or a 23andMe or a non-profit. If we're getting some value out of that, I think we have to, in some ways, give up some of our own kind of privilege to protect some of that data. But that fine line is different for every single person in the room, right? I might participate in 23andMe or not Ancestry or not or other ways around, right? Ancestry might know less about my health, but more about the ancestral patterns. 23 might, if I don't answer questions or I do, they know what they know, right?
Yeah.
So it's a tough world. I think everyone is going to end up facing it themselves. Facebook is another example of many of these, right? This big data world.
Right, and it doesn't just affect you, right? Like it's not, like this is the part that was really striking to me.
Yeah.
Because I had thought a lot about like, okay, what is my own genetic privacy like? Like what am I comfortable with? What am I not comfortable with? But I hadn't, like it just hadn't, the penny hadn't dropped, right? That it was potentially like, not just mine, but like my parents, and my siblings, any potential children, all of that.
That's right. But you could argue geolocation the same way. You tend to live with your family members. You go to school with your schoolmates. So there are these associations. Genetics might be a little bit more direct, or at least seems more biologic in some ways. But it doesn't have to be just that one in that way, right? Now, I know a lot of people are on Facebook and they've seen some of these kinds of reports. I remember, again, I'm not saying one way or the other here, but one person noted that if his family member was indeed the killer, he would have no problem giving up that family member to the police, right? So again, you can all decide, everyone's going to decide where they stand on that.
Right.
But there is that other side of it that why would you want to harbor that person necessarily?
Sure, and I mean, I guess that sort of brings us to the other part of DNA, which is, of course, that there is a medical application, and that there's some. It's not quite where we thought it would be, let's say 10 years ago.
Yeah.
But it's certainly coming along.
Yeah.
So maybe you should start by like just letting people know what precision medicine is and what it's got to do with DNA.
Yeah, sure. Okay, so let's just think about for a second, why isn't DNA where we are, right? I mean, we were hopeful for a lot more. It turns out those other pesky things like exercise and diet actually have a lot more to do with disease than your DNA, right?
Weird.
Your DNA, would you imagine that? Your DNA is your DNA from birth, right? With some exceptions like cancer and the rest. You know, you think about it, you're born with that DNA, and somehow we're thinking that's going to predict what disease you're going to have on the order of 100 years, right? And even the databases we can compare to are changing out from under us. The DNA is not a fortune teller in that way, right? A lot changes based on what you eat, what you exercise, and those variables and connecting them to DNA are making it really tough, which is why we have this field of precision medicine, hopefully to try to sort all this out here, right?
I'm gonna have you hold that thought because we're gonna talk a little bit about another medical use of DNA on somebody who has not been with us for quite some time.
That's right.
So you were involved with this DNA testing of a Chilean mummy.
That's right.
Who was frequently rumored to be of alien origin. Really, this was a thing. And so to settle the question, you were among a group of scientists who did some DNA work to see what was going on with this mummy.
Yeah.
So maybe what brought you to study the DNA? Was it really just one of those things where you're like, this can't possibly be an alien? Somebody's wrong on the internet. I gotta do something about it, or was there, another motivation?
Yeah. So first of all, this work was done by an enormous team of people at UCSF and Stanford, and my good friend and collaborator Gary Nolan is the kind of alien skeptic of the set. He's got involved with several kinds of projects to try to debunk anti-science kinds of folks, and this is one of those here. So this is a mummy that was really touted in Chile for 20 years. Now, there's an unfortunate nature to this story, right? This is a skeleton. This is we know now is a girl, probably preterm, so probably was never actually born, and had these unusual mutations. Now, part of my work is when I look at DNA, one of the things that we can try to do with that is try to figure out why current day patients have these kinds of problems, or what in the DNA is connecting them to their symptoms. So I'm in pediatrics. That field is revolutionized. It used to be that you could have a child and we wouldn't know for decades what is this syndrome causing these weird abnormalities, whether it's something we can see or lab tests or a child is having fevers or infections. We wouldn't know. And then you get a learned scientist and maybe find 100 of these kids and maybe you'd write a syndrome with your name on it. And then we got to 10 kids we could figure this out. Now we can do this with just one. With just one child that comes to, I'll give credit to either Stanford or UCSF now, we can sequence the DNA and figure out which of those genes have a really abnormal spelling that's leading to a syndrome. So we've done this for patients and kids. I naturally thought it would be interesting to do it for this particular patient here, who was already deceased here.
Sure.
And so in this way, we were looking for mutations in some of the bone, we were looking for mutations all over. We found many mutations in bone genes that had never been seen before. We think that's what would explain the unusual signs that we saw here. And they could probably, we think, why would I get involved in this, is to build on the database, so then the next time a kid actually shows up to any of our children's hospitals, that we know we can compare the database and say, this is explaining that. That you parents have an answer here, right? Instead of a diagnostic odyssey that goes on for decades, actually.
Well, DNA is increasingly being used in archaeology.
That's right.
Is there anything that we can learn from that kind of past? I mean, obviously, when you mention mutations we haven't seen before, the possibility of being able to use the knowledge from the past for the current day.
That's right.
But I mean, I think most of us are curious about where we come from. What are some other potential uses for DNA in archaeology?
Yeah, so I think in archaeology, certainly, and many articles have written about this, controversial ones too, that the use of DNA is actually really exploding right now. The first thing I got to really say is everyone, and we and everyone else, have to really do this kind of science in a respectful manner, hands down. I mean, there are cultures, there are populations that don't necessarily want to know more detail about where they come from. I think that has to be respectful, especially acquisition of samples. All of that has to be done in proper ways. I think there are certain cultures, certain subpopulations that are higher risk for diseases than others. Certainly obvious ones like skin cancer has a lot to do with, for example, skin color and things like that. But that's just one of many of these kinds of examples. So I think in the future, some of those might be attributable to ancestry, to human migration. I think maybe we'll get better explanations why we have these diseases in humanity. Right? Why do we have interesting or weird diseases like cystic fibrosis? We have some ideas now, right? Maybe that protects against cholera in some forms, or sickle cell malaria. We have many of these kinds of examples. But I think in some ways we learn more about the biology of a disease and maybe get a little bit closer to a therapy someday.
What did you most take away from this work on this particular mummy? What was the thing that really stuck with you, having done this work?
So I'll give you the science answer first, right? Please. So the scientific answer, what we think is happening in this particular child was that there were more than one mutations happening here. Now, why that's interesting is we're believers of something called Occam's razor, right? Which is the kind of belief that goes back that the simplest explanation is often the right one. And so when we do have patients, these kids come in with these syndromes, we're typically looking for the one mutation that could explain it all. And just to make it kind of more concrete, when a kid comes in and we sequence a child and maybe we get both parents, there's still about 50 differences in that child compared to the parents. That's just normal DNA mutation. Not all of those differences are in the recipes. It could be in the table of contents. It could be in the index, right?
Maybe the binding.
In the binding. There's a lot of binding in the DNA, right? All those things, those folds. So there are a lot of these differences that's normal mutation. And then we're trying to figure out, well, which one of these led to this syndrome? Well, this one is in these recipes, and these recipes seem to show up in the same bones and tissues that this disease seems to be in, so maybe it's that one. What I learned in this case is that there might be more than one of these mutations there, right? There could be, it is possible to have kids that actually more than one that are causing the problem. That's what we think happened here. Now, in this particular case, I think this is obviously a controversial sample. Where the controversy are the two extremes, where, I'll be honest with you, we have on one extreme, there are people on YouTube, I'm not gonna tell you to go look for it, but you can see them, that still believe this is an alien, and that we were influenced by the government. I don't know, those NIH grants are doing something. And they literally
you would need a lot more money than the NIH
and to
potentially
be
could give
super
you.
clear, we use no NIH money for this, okay? We have enough money at Stanford, UCSF, and to just do this kind of work, right? So no government was involved here, okay? But we have that extreme, right? And then the other extreme where there's a group of folks which are legitimately concerned about samples leaving their country, the illegality and legality of this, that are now wondering, well, why was this sample bought and sold so many times in Chile? Where did the sample come from? And all of that. So there's these two extremes here. I thought we were helping to figure out some mutations that might help other kids in the future. That's why I got involved.
See, this is the thing that I like, though, because it touches on something. Science is never happening in isolation. Medicine is never happening in isolation. It is, of course, happening with people.
That's right.
And all of the things that come along with people everywhere else also come along in medicine.
Absolutely. I mean, there is a lot of context. I mean, that is exactly that. I think every country is different. Every state in the union is different in how we deal with these types of issues. And it all shows up at the same time on Twitter nowadays, right?
That's right. It's context collapse within the context of no context.
That's right.
Which sort of brings me to the more clinical context, which is precision medicine, which we hinted at a little bit earlier. Maybe I can get you to give me a more specific definition.
All right, so the definition of precision medicine, there's official ones and all the rest. To me, precision medicine is the customization of healthcare, okay? We're gonna customize the healthcare we deliver to you based on your measurements. Some of them are gonna be biological measurements, like your DNA and proteins and things like that. But it's more than biology. A lot's gonna have to do with your exercise, your behaviors. I might get them from smartphones or wearables. A lot's gonna have to deal with your preferences. I can tolerate these kinds of side effects. I don't want that kind of side effect from a drug, right? So I gotta get a lot of data from you. Some of it's biology, but not all of it, your environment and all that. But I can't even just do it with your data. I also need similar measurements from a lot of people, and even more importantly, figure out what medical care did I give all these people, and what's working and not working, and then apply it all to you, right? So it's more than just personalized. The same thing about context. I can't just do it with your measurements. I gotta compare you to many others and figure out of all those we've treated before, what's actually working and not working. That's the hard part.
That sounds like a staggering amount of data.
It's a lot of data, and we gotta get more of it. I mean, we have federal initiatives which we can talk about, but you know, I think we don't have enough, and we always want more. A data guy's always going to say we need more data, right?
That is generally what they do.
That's
they
right.
love having their jobs. They want to have more jobs in the future. They
Yeah,
need more data.
Yeah, there's certainly a lot of job protection in the data sciences right
That's
now,
right.
especially in the medical world too. I think we are drowning a lot of data, and we have these efforts now that are out there that we're going to be getting more.
So just to give us a concrete example of some things that have come out of precision medicine, are there specific therapies? Are there things like that we should be sort of thinking of as examples?
Yeah, so the one area where precision medicine's really had the most traction, I guess you could say, is in cancer. So we have drugs that are targeted therapeutics now. So for example, one particular drug for melanoma, vemurafenib, it's only really used for malignant melanoma with a very specific DNA mutation. So that means now the doctor or the oncologist or a skin doctor will have to take that sample of cancer, run it in a sequencer in just the right parts, and if that mutation's there, then they're supposed to be using this particular drug. That is as hyper-targeted as you can get. It's got to have that mutation. So that's one of many such examples. I think last year the FDA approved on order of 30 or 40 of these types of therapeutics. Now, that in itself is going to be challenging because what if you don't have that mutation, right? I mean, so that's going to be something we can definitely chat about. But I think we are getting to more of these precision types of therapeutics. One of the earlier ones was from a company called Genentech just up the road. Herceptin is a breast cancer drug. That's an example of a drug that would fail if used on all breast cancer patients. But actually, when they realized it was working on a particular subset that they didn't know about, and then they tested in just that subset, it had great efficacy. And so what another part of precision medicine isn't just thinking about diseases the old way, but diseases the new way here.
And so when you say thinking about diseases the new way, I'm just going to press you on that a little.
Yeah.
I think what you mean, and stop me if I'm putting words in your mouth, but you're talking about, thinking about breast cancer not just as breast cancer, but as like sort of a larger designation within which there are many different kinds of cancers with certain kinds of mutations, like HER2, those sorts of things.
Absolutely. And Liz, I have some slides on this.
Yes!
So now we hit the right keyword. All right, so precision medicine. So the word that I'm trying to use here, that I'm going to teach you, is this word called nosology. Nosology has nothing to do with your nose. It's the systematic classification of diseases. The pigeonholes that we put all these diseases into. It goes back to this guy. This is Linnaeus. So some of you might remember in grade school, you knew Linnaeus, or you at least had to memorize at one point on a test, kingdom, phylum, class, order, family, genus, species. I see some of you nodding. You all had mnemonics for this. Some were clean, some were dirty. Don't repeat them here. All of you had them on a test at one point, right? But Linnaeus is also the guy who put diseases into a taxonomical tree. So you can get these books from Google Books, and this one's called the Genera Morborum, where you can see they were trying to use fonts on the right there, making a tree of diseases. Now, he's not remembered for this because he got it horribly wrong, right? Why remember the guy for this when he got this species so correct, right? But basically what this tree says there is put the fevers over here, but fevers with urinary pain over there, and fevers with skin problems over here. Everything was fever, 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 right? That's the best you could do. And mind you, this was about 90 years after Galileo was under arrest for saying the sun was the center of the solar system. So it was pretty good news.
So,
right?
so it was well before we'd figured out anything about bacteria.
Absolutely. But the thing to do, way before bacteria and microscopes, but the thing to do at the time was, if you were a learned physician or a philosopher, you got to make a new classification of diseases.
You also had to do it in Latin.
Yeah, this is Latin, and again, font sizes and all the rest. This is printing press era here, right?
Wow.
So then up to, let's see if it goes to the next slide here. Yeah, so fast forward about 150 years, up to 1860, William Farr and the Royal College of Physicians came up with now what we call the International Classification of Diseases. Okay, and so this is now what we use as ICD-9 and 10, which we'll talk about in a second. So the current classification we use now just started right after the American Civil War. Okay, that's how bad it is. So this is ICD-2. And in fact, I have a copy of this book here, and I literally carry this everywhere, ready to go. So this is ICD-2. It's pretty thin here. And Manual of the International List of Causes of Death, second revision, Paris, 1909. So just for the history buffs for a second, this was not a WHO standard because, of course, this predates the United Nations. So this became a League of Nations standards, if you remember your European history, right? And this book is amazing, okay? Because it's written the same way our code books are written. And I'll zoom in here to make it easier so you don't have to read this. Yeah, I'm supposed to hold it in this hand here so the camera gets it for Facebook. So it's written code 39. It says it's cancer of the buccal cavity. That's cancer of the cheek. And it says code 40 is cancer of stomach and liver, cancer of the intestines, cancer of the skin, and it's amazing. Okay, 100 years ago, they did 100 years ago what we still do today, and this is our problem. We're running out of numbers. Put all the other cancers in 45. Okay. Oh, just put all the rest. Cancer, not otherwise specified, right? And I love this phrase, not otherwise specified, because it's all the kind of annoying, embarrassing things in medicine, that they're not worth distinguishing from each other. And literally 100 years ago, this book proves it was not worth distinguishing lung cancer from brain cancer, right? You couldn't get x-rays. No one looked in a skull. Oh, it's sort of the same thing. You can't do anything anyway, literally, right?
You're stuck.
Yeah, we're stuck. But we still use not otherwise specified. It's all over our code books. And here's where it gets even funnier. This book literally says that if you run into a patient that died because of a visitation from God, be sure to use code 189. Okay?
Is that code still in use?
You don't get paid much for that one. But like, can you imagine the same way you're laughing at this, right? Can you imagine how they're going to be laughing at us in 20 years? Right? Those silly fools, this joint disease and this bowel disease are the same thing, right? That's how they're going to be laughing at us. And so the narrative I want to try to tell you is we got to get away from books like that to, oh yeah, okay, this one. We've moved on to ICD. We moved on to ICD-10 now, which is the 10th edition. A lot of memes and physicians criticizing. This is the one that I like. We have codes for everything now, seemingly. I don't always get sucked into a jet engine, but when I do, I use ICD-10 code V97.33XD. So it's literally a code if you're an astronaut hit by a micrometeorite, if you're a surfer hit by a whale, and physicians make fun of this. Now, so I've been to San Diego many times. And in San Diego, there's a great naval base there, and they're aircraft carriers. And indeed, they've told me it is possible to get sucked into a jet engine. Okay, it is possible. But this is the chronic form of the disease. So I can imagine getting sucked in once. How do you get sucked in twice, right? But if you do, there's a code waiting for you, right? Okay, so we physicians make fun of ICD-10 because of all the codes, but this is what we should be thinking about, right? But actually what I'm going to kind of leave this segment with is ICD-9, which is what we had for 30 years, these codes. What do I mean by these codes? If you ever go to a doctor that gave you a sheet to hand back to the receptionist, the codes were in the back. You saw the grid of codes, right? So we go to the next slide here. This thing, let's see here. Yeah, here's ICD-9. Here's lung cancer again. So if you have lung cancer in the ICD-9 world, your code is 162.9. And here's the problem here, right? So here's all of lung cancer. You see all the listed diseases on the right there. Did we take into account the latest in gene sequencing to figure out which lung cancer is which? No, of course not. Did we take into account the latest molecular testing to know this lung cancer or that lung cancer? No, of course not. Did we even take into account the basic microscope where we can tell large cell cancer from small cell cancer? No, we did not. You know how lung cancer is coded in ICD-9 and ICD-10? Left lung or right lung, right? It's literally that, right? So we are at ICD-10. We've had it for 2 years. It's not the end. It's the beginning. We got to be more precise about telling patients, other doctors, and now the computer what we think our patients have, so the computers can help us with all this. That's what we got to get to here.
I see, I see. So when we're talking about, let's say lung cancer, just because that's where we are, maybe it is more helpful, instead of saying lung cancer, to start thinking about the genetic components.
Absolutely.
And maybe some of the stuff that shows up on the microscope.
Absolutely, microscope or microarray, right? So now we have the narrative to think about here is to replace books like this with cool gadgets like this. So this is a DNA chip. I think the next slide, if you go to that one, has a picture. Yeah, okay, there you go. So I picked this one, Affymetrix. This is how I got my start 20 years ago. Big data comes from little packages, just like this, right? That's why, and data is ethereal, so I carry these around in my bag. I kid you not. And Affymetrix is down in Santa Clara. It's a great company. It's how I got my start. You can put a sample of cancer or biopsy or cells in a petri dish or animal models. These were priceless 20 years ago. Now you got to read out the entire genome for a couple $100. I literally carry this one in every suit. And what's amazing is everyone uses these. It's not a big deal anymore. And then we get tired of measuring them one by one. And then we switch to the 96-well plate format. And that's so cheap now, I carry that in my bag. And then we got tired of measuring them 96 at a time. And then we switched to the 384-well plate. And that's so cheap now, I carry that in my bag.
So if
I
you need
cannot.
a sample, you know who to go to.
You need samples, you need props. If I cannot more clearly illustrate exponential growth in biomedical data than to show you these plates, right? And we're used to this, right? We see chip wafers and things like that. This is my example of that. We don't even use this one anymore because we use sequencers of this kind of science. Affymetrix got acquired 2 years ago. They don't even exist anymore. They got a new name, Thermo Fisher. We just keep making more and more of these measurements in biomedicine. And the whole idea is let's get rid of this kind of thinking, replace it with this kind of thinking.
So what have we learned from the early efforts in this space? I mean, obviously we've had to sort of stumble along as we always do, inventing stuff, figuring out that it's wrong, and like correcting it, and all of that. So what has that process been like? Yeah,
so that is the process of research. We don't always know the answers. We know some of the questions we want to ask, and sometimes we get some answers, sometimes we don't. That is what research is, especially in the medical world. Some of you may have conditions. Some of you may have been asked to participate in trials to test out a new drug or not. And without doing those kinds of studies, we'll never learn what works or not. And this particular world, we're trying to study the hard diseases. We have a lot of hard diseases. We have neurological conditions. We have cardiovascular disease. We have diabetes and all of its complications, and many other disorders where we still don't know why do patients get these diseases, and what else can we use to kind of target those for drugs and other therapeutic interventions. So there's a long way to go in medicine.
One of the things that I'm curious about, though, is precision medicine, obviously powered by patient data.
That's
no
right.
other way it can possibly work. So in terms of unforeseen consequences or potential risks, how should we be thinking about this? Yeah,
so in some ways, it's the same story that we even started with, right? To give, you're going to get, right? I think we don't know what we are going to find yet, right? How is it the process that patients get diseases like diabetes and Alzheimer's decades long, right? It's hard to study decades-long processes in any field.
Also expensive.
Yeah, well, ask the climate change scientists and all the rest, right? People don't believe and trust because it's not happening in front of our eyes. And some of these are so gradual processes that we need ways to really get measurements from patients, from participants, as they get into these diseases. And that's what some of these new efforts are around the country, to try to get to follow patients for a long time.
So in terms of thinking about privacy, and I'm just thinking about myself here right now, very selfishly. If I wanted to think about how best to approach precision medicine while also trying to safeguard some of my privacy.
That's right.
How would I go about doing that?
So first of all, I think privacy is now on everyone's minds because everyone simultaneously is getting an email today from GDPR, okay? And so the anti-spam legislation is generating so much spam, which is ironic today. So everyone's hearing about the European privacy rules. and how now they're going to be emailing us before they email us, which I still don't get. But we're supposed to think about privacy. If you're a company that does any, or any organization that does any dealings with Europe, we're supposed to think about privacy. Now, when I hear, again, I'm not a lawyer, but when I look at GDPR and all the kind of summaries, what I realize is GDPR is now putting on other data where we were decades ago in medicine and health, right? So we've had HIPAA in the United States. Now, HIPAA to some people is a curse word because it slows them down, and that's not the way to think about HIPAA. First of all, HIPAA has a lot to do with billing, right? We got to use the right codes, we got to all agree to certain codes and things like that. But in some ways, it made consistent across the country the rules to get to this kind of data, right? And so when I hear about what GDPR is like, it's actually a lot like where we were a long time ago. So for example, there are safe, responsible, respectful ways to use this kind of data. That's what we all believe in. We don't want to do this in any other way. And there's federal law that punishes us if we do the wrong thing, and it's always been there like that. So if we want to do things with de-identified data, it's a particular phrase I use a lot, where we strip out identifiers in a legal way, there's federal law. It's in federal law how to de-identify medical record data. If we then need some dates, or if we need some names, we need to get institutional review board or ethics board approvals, and a lot of eyes on what we do to make sure that it's safe and respectful. So I think everyone's thinking about privacy now, but I think in health data, we've been there for a long time, actually.
Which sort of leads me to my next question.
Yeah.
Because obviously there's the whole genome sequencing, and then you have sort of specific tests for things like HER2 or BRCA or KRAS, depending. How do you think about when to use the whole genome test versus when to use a specific test?
That's a great question. When do you use which test? And to be completely transparent, nobody really knows the right answers.
Great,
right?
Love those questions.
Yeah, so the world of diagnostics is moving pretty fast now, right? So let's find a really easy extreme case. If you're a parent of a child, you don't know what's going on with the child, it seems like something genetic. The old world, and old world means only 5 years ago, we might try one test and another test, looking at one recipe at a time. I think people have shown now cost effectively, just get the whole recipe book sequenced, figure this out. It's much obviously cost effective, even just in terms of the doctor's visits. And the phrase we use is diagnostic odyssey, right? That gets shortened there. And that's taken to an extreme now. Rady Children's down in San Diego, they have literally the Guinness World Book of Records time that they can go from a newborn to a genome sequence. I think their record now is 26 hours, okay? Now remember, compare that to 13 years and $3 billion, and that was just 2003, and now we're down to 26 hours, okay, to a genome sequence. Because sometimes you need to know right away, what are you going to do with this baby that is not doing well, right? So that's an easy extreme, right? Where it gets harder are things where, if you look at genomes, you might find spelling differences that you're not sure are going to make a difference or not. And so take examples, a controversial example, not a genome one, but prostate-specific antigen, or PSA. So PSA is a well-known test a lot of doctors use to predict or to tell if you're having prostate cancer or not. But it doesn't necessarily tell you if you're going to die of prostate cancer, right? You may have other things that get you before the prostate cancer does because they're slow-growing cancers. So then you're stuck with a problem. What do I do with this cancer? Is it going to kill me, or am I going to outlive it, right? Those are the challenges that we don't know sometimes. If you run this test, what's the right answer? And we can do studies and go back 10 years and figure out what happened 10 years after. But the environment keeps changing out from under us as well. So those are tough studies to run. That's exactly why we need to start thinking about very long longitudinal studies in people to figure out what happens next for some of these sequences.
Well, this is an interesting thing, though, because obviously part of what has made it much easier to sequence DNA has to do with technological advances,
Absolutely.
better computing power, all of those things. And so when you're thinking about these long-term studies, obviously the technology will be changing under you. It's not going to be like one of those things where you make a NASA probe and you have what is essentially a PS2 on it, and that PlayStation computer is sending you back, pictures from Pluto all this time later. How do you design longitudinal things in such a way that they manage to keep pace with the technological advances?
Yeah, so it turns out with DNA, at a certain point, we're going to kind of hit a kind of threshold where when you have a DNA sequence, at some point we're going to believe it, and we're not going to get much better. Now, where the kind of devil in the details are, sometimes DNA is different in different tissues. Okay, so that's a new one, right? Part of my body might have slightly different DNA than others, depending on how you were formed in the beginning. So there might be subtleties there. And different cells have different DNA. So for example, your immune cells, they chop out a lot of parts of DNA to make the right antibodies and things like that. So we might go into more detail, and cancers or all the rest might change DNA. But in general, your basic DNA, or what we call your germline, when we have that, we're probably going to be confident we have that. The other side of life, everything but the DNA, changes constantly, right? For example, our diet and environment changes. And if any of you don't believe that, we do have a range of ages here. Many of you do remember what life was like before the Cheesecake Factory, right? Before 1,000 channels of TV, right? Before, before we thought it was unsafe for kids to play outside, right? Those things changed in our culture way faster than the DNA did, right? Today, I told you, 50 changes maybe per generation, maybe, right? So that stuff changes so fast. I think we're going to get better ideas of that with social media, apps that are going to be tied towards research, where you're entering in or taking pictures of food. A lot of people take pictures of food now. Stuff like that are going to make a world of difference, I think, for research.
You said something interesting that I want to come back to, which is that DNA varies in different parts of the body, which is true. It's especially true, by the way, if you observed your twin in the womb.
That's right.
Which a lot of people do.
It does happen.
Yep. But one of the things that I'm curious about is specifically when that comes to certain kinds of diseases, the one that immediately comes to mind in cancer, often there are mutations in the cancer that are different from the rest of your body.
That's right.
How should we be thinking about that?
Yeah, so cancer testing is obviously the biggest area where another great example where we might go from 1 or 2 genes to now getting hundreds of genes. And so companies in California and all over the country now are involved with sequencing DNA. If you are a patient with cancer, let's say a harder cancer or later stage cancer, there are chances that it might get sequenced either by a hospital system around here or one of these kinds of companies. So that DNA sequence is there, and what we're looking for, where is this cancer different? And specifically, where is it different? Because cancer is really a disease of mutations. One of the earliest genes to have a mutation are the genes that control the growth of that cancer, right? So the minute you knocked out the brakes, you know you're on a ride, right? And then another set of genes to fix DNA, the DNA correcting part, you knock those out too, so then you get even more mutations. And it just keeps building. It's a positive feedback loop, and those are really tough. It's really tough to shut off positive feedback loops in any industry, not just medicine. And so in those kinds of examples, we have a lot of those measurements, and we also compare them to the regular DNA or the germline to see where they're different. And why we do that, especially in kids, is even if we see you have a cancer and we're trying to figure out what's wrong with the cancer, maybe we can treat it, it's also helpful to know, are you set up to get another cancer? Right? So you might have shown up with this cancer, but you actually have something wrong or different with the rest of your DNA. We better make sure you're not going to get another one too, right, later on.
Of course.
So we're going to be even more careful. So that's why we also look at the germline or the regular DNA as well.
Well, here's a thing that I am curious about. Obviously, nothing is perfect.
Yeah.
And there are sometimes mistakes made with DNA. For instance, in the case of the Golden State Killers we were talking about earlier, there were some false matches.
Yeah.
How should we think about perfecting the technology that makes this possible and any kind of uncertainty that comes with it?
Yeah, so there are some hard parts to get to at DNA. There's certain things that, certain spots of the chromosomes that are really tough to sequence. And so how does sequencing actually work? Let's just talk about that one for a second. So it used to be, the conventional way was literally, sequencing through Jello, okay, literally Jello. And so what we used to do is, humor me for a minute here, since we have more than a minute here.
Oh, yeah.
The old way to sequence DNA was to think of a copier, right? So you remember copiers, nobody prints anything anymore. My parents still print things. I'm sure some of you do too. But we make copies of a lot of things. And so what happens when you're in a rush and you're making a copy? The copier jams, right? Everyone's used to that. Sequencing of the old was making the copier jam. And nothing makes copiers jam more than fiddling with the paper, right? Like making thin paper, thick paper, fluorescent paper. Let's put fluorescent paper in there. That's literally what sequencing was. Get the copier, the body's copier, artificial copiers, to jam with the fluorescent pages and figure out what letter was next to that. There are only 4 letters, A, G, T, C, 4 kinds of pages. And you just basically run it on Jello and literally see all these kind of readouts there. That's the old way. You could get to a couple hundred base pairs at a time. Remember, 3 billion, couple hundred at a time. And literally, you use computers to stitch the whole thing back. So think of it like a puzzle. You've all done puzzles. You see the edges. You don't know what the finished picture looks like. And by the way, half the pieces look exactly the same.
Love that.
Because there are all these repeated elements again and again. So that's the old way. The new way is to cut DNA in even smaller pieces, but get it sequenced in parallel. And yes, computers have made a difference. The speed of processors have made a difference, but nothing has made a difference more than actually higher megapixel cameras, it turns out.
Really?
Yeah, because these little fluorescent flashes now are so fast and so tiny that with higher megapixel cameras, you can get more of them done in parallel, and that's actually made a world of a difference too.
That's wild. I would not have guessed that.
Yeah, so that those sequences now really are taking pictures like crazy, and literally they're flashing different colors if there's an A going or a T or a C or a G. And so you got to amplify, make a little bit more, and then sequence. Sometimes errors happen that way, but now we're getting to even single molecule sequences with just the same DNA with no amplification. It can go right through here. So that's basic sequencing technology. There are still problems with it. Repeated elements. If you have an A like in a row, were there 7 of them or 6 of them? Couldn't really tell. Those kinds of errors. Repeated elements cause it to skip. You just remember records used to skip. Kind of like that, it gets back to where it was. It's not where it's supposed to be. Those kind of errors happen in sequencing as well. But nothing is worse than putting the barcode on the wrong tube, okay?
Oh my God.
So that kind of human error, we have to watch out for, right? And so we have standards for that. So in the United States, we have one easier standard called CLIA, Clinical Laboratory, a whole bunch of questions coming. Look at that. CLIA, I cannot remember what it stands for now, but that's how we actually test the quality of barcoding and make sure no mistakes are being made through CLIA so that they can bill for tests. And the higher standard is called CAP, the College of American Pathologists gives that kind of blessing, and that's another, it takes a couple of years for a lab to reach that level. And then the FDA might get involved if you're selling kits. Otherwise, they don't really regulate right now. That itself is controversial. Lab-developed tests, they don't regulate that at all. These other two bodies regulate those. That it's legitimate and it's right for billing. You can get paid for the work here.
So here's a question. There's already a trial, a CRISPR trial going on in the US. There's another one in China. For those of you who don't know, CRISPR is a specific way of altering DNA. It's like, I tend to think of it as like copy-paste, right? Where like you snip out a part of the DNA and then you paste in something new. And the person who's doing it in the US is Carl June. Who is an immunologist who has successfully gotten the immune system to fight certain kinds of blood cancer.
That's right.
So I guess what I'm wondering is, obviously these two things seem like they would sort of intersect.
Yeah.
How do you see that coming to pass?
Yeah, so CRISPR is great, and it's going to be ways to edit and copy and paste or fix certain spellings here and there. But it's not going to be that simple for a lot of things. So cancers, again, are a little bit easier, especially blood cancers, where we get the tissue, you could take it out, maybe fix it, maybe put cells back. Those are going to be easier. It's much harder to try to fix a spelling error in every cell in your body. Right? We have no delivery mechanism at all for that. Now we can hijack how viruses spread things over our body, and maybe you put those kinds of gadgets in those viruses, and so people do that for lung diseases. Think about, maybe you can at least get all the lung cells this way, or maybe all the liver cells this way, because viruses attack the liver. But otherwise, to fix a lot of these, the delivery part is going to be the hard part still, no doubt. And then we still don't know about off-target effects here, right? Maybe we think it's fixing one thing, but maybe it's changing others too. We don't know about those yet.
Yeah. So what has been the biggest surprise for you so far in genetic medicine when you're thinking about, this entire very broad field? Have there been any moments where you're like, gee, I didn't see that one coming?
Yeah, so, probably a lot of surprises. If you couldn't tell, I'm kind of really excited about this field. Couple surprises. First of all, it's amazing how much kids love this field, okay? And so just a shout out to the Bay Area Science Museums for a second, including the one you're in now. You go around the country and a lot of people say, oh, this is a great place to raise kids. Nothing beats raising kids here, okay? And what do I mean by that? For example, when our own daughter was 3 years old, we took her to the tech museum. And even at age 3, she was taught there because they put on gloves and they watch a video to put on goggles. And they literally were taking jellyfish glowing proteins and putting them into bacteria, right? And one morning she comes in our bed and she says, I gotta go to see if my experiment worked in the lab, right? Like 3, 4-year-olds can do this. Only in the Bay Area can we do that. And now it's synthetic bio there, right? I mean, so it's amazing that this community gets it here. And we're embracing it. We're trying to teach our kids how to do this. That's super important here. But it's also the rest of the public. I mean, how much DNA sequencing has really captured the public. And by this, I got to give credit to 23andMe and Ancestry, right? Where millions of people have actually given DNA to try to learn more about themselves. That's a complete shock to me, that it's just really, that people see that that's important and it's valuable, and they want to learn more, and they're going to get more out of DNA.
Well, this leads actually to an audience question.
Okay.
Which is, what are your thoughts on direct-to-consumer genetic testing in the health space? And I feel like are you misremembering that you're involved with Helix?
Yeah, so I have conflicts of interest up the wazoo here,
Just
okay?
FYI.
So all of these things, right? I'm involved in a lot of companies. So there are several direct-to-consumer companies. I'm involved with one called Helix, which is a little bit more on the sequencing side, but they also make socks, right? That you can have socks made with your DNA patterns. So every extreme, right? And so there are every extreme of things you can do with your DNA. That's just one example of many of these where people really are enamored. They're partnered with National Geographic, same kind of Ancestry product. Most people have gone in that way, it seems. So thousands of people, millions of people are really trying to learn about DNA that way. I have nothing against those companies at all. I think that is the future. Look, we're not protectionist in medicine. I mean, some are, but this is, how can you say in the end, people learning more about their body themselves is a bad thing? Now, where there is some concern is where this happens, where you see something and then you get worried about it excessively, right? Now, people have run tests to see whether depression, anxiety goes up after genetic testing. It doesn't seem to. But let me make an obvious point a little bit more obvious. If you got a 1 in a million disease, right? 1 in a million. And now I tell you, you're 1,000 times more likely to get that disease. It's still 1 in 10,000 you're going to get that disease. It doesn't mean you're going to get it, right? Even if I say you're 1,000 times more likely to get it, it was still super rare. It is still super rare, right? And so one of the hardest things that I learned, another surprise, is how terribly we all, myself included, understand what risk means, right? What should we be worried about? You've all seen the internet memes of what you should be worried about. If you have any kind of want proof of that, just go to SFO and just look at security theater and all the rest of it. Right? And that's just one example of many where we don't know what we should be worried about. Right? The last thing on Twitter or on CNN seems to make us more worried than the real things we should be worried about, myself included. I don't know how to fix that. We need more data viz. We need more risk viz. Ways to communicate risk to people, patients, and physicians. We just don't know how to do it.
I gotta say, the older I get, the more I wish I knew more about probability, because I feel like that would be a really nice way to reassure myself. But no odds, I
know. So odds, but do you use odds or likelihood ratio? Or there are all these statistics which are all valid, and they all kind of give you a different answer. Do I put 100 people and show one of them in red, or how do I show that across time, across age? And so to make an obvious point obvious, we're all going to die someday, right? We are all going to die. We haven't figured that out. Well, I don't know what you're investing in. There are companies that say not, but I think we're all going to die. And actually, in the United States, it's illegal to die without a disease, right? We must put something on your death certificate, okay? This is illegal, okay? There must be an ICD-10 code on your death certificate. So not only are you going to die, you're going to die of something, right? And so can I fine-tune it? Or, maybe... I don't know the exact percentages, but is it cardiovascular? Is it brain? Is it your heart? Is it cancer? Is one of those 3 big buckets? Maybe that'll fine-tune things. Maybe you can precisely prevent in that type of way. Of course, get all your vaccines and stuff, but maybe I can fine-tune what I could do to. But in the end, when people ask me about this in genetic testing, one question I've been asking more people more recently is, what do you want to die of? Right? What do you want to die of? And not to be morbid, but it is also something we often want to die in our sleep, right? Everyone says that, right? And you want to cliff the disease. I'm great, I'm dead, right?
Yeah, that's right.
I don't know what disease that is anymore, right? That used to be the heart, but I'm not sure if we're fixing that one. I don't know what that's going to be. So I think about these things, not in a morbid way, but that is life, right? That's part of life. And so we don't know how to communicate or to understand risk.
Well, there's a sort of a second part of this question, which is actually not from the audience and just is a question of mine.
Your prerogative, of course.
It happens. But one of the things that I think is particularly difficult is that frequently, if you have gotten your own DNA sequenced and you bring it into your doctor, your doctor looks at you and says, "That's very nice."
Yeah, well, doctor, am I going to get these diseases? And by the way, the nurse says, doctor, your next patient's already in the waiting room, right?
So how do we figure out how to communicate about these things with our own clinicians? I think a lot of folks who, especially ones who haven't been through medical school very recently, maybe don't necessarily know how to deal with DNA in this way.
So first of all, the future of all this kind of DNA-based device is clearly computers, hands down, right? It's impossible for medical professionals to stay on top of everything in every field. That's a given. So the computers have to get better. We were talking in the green room about electronic health records right now. And right now, if you've been to any doctor recently, you know what an electronic health record is because you've seen the back of some doctor's head as they're typing into a computer. And it looks like Windows 95, it probably is, and we've moved on. You know what I'm talking about here. It is really that old. And so these computer systems, and they're horrendously expensive, but we're using them. But right now, still, doctors, kicking and screaming, are entering in the records as they're seeing you or after the visits. What we have to change to is a doctor that is going to use the computer before you come in to get the advice. What should I tell this patient? What is going on? We're not there yet, right? That's called decision support. They barely want to use it after the encounter. We got to now switch to before the encounter. That's a long way to go. But the computers are going to have to give the advice. And so my future vision for DNA, we had this really unusual patient, Steve Quake, who was a professor at Stanford. He was one of the first patients to have his DNA sequenced as a patient. And what I learned from that experience is that the future of fixing your DNA problems is behavior and environment, right? Behavior and environment is the new prescription pad, right? So what if I tell you, well, you're doing great on lifestyle and stress, but you're still a smoker, and here your DNA is suggesting you are at more risk than most to get lung cancer. Let's work on this particular thing the next 6 months. Will that make you more likely to quit or not? I don't know. But it's a hypothesis we have to test now, right? Does giving advice with your DNA make you more likely to change or not? We don't know the answer. I think we're going to learn as we go in the near future.
This is an audience question, which I think I maybe know the answer to. And it is, can we patent personal DNA? And I'm pretty sure the answer is no.
Can we patent personal DNA? Okay, so again, what do we say on the internet? I'm not a lawyer, right? You know the abbreviation for that. So first of all, and there are probably patent folks in the audience here, so I'm not going to misspeak here. So first of all, a lot has changed in the patent world recently, okay? Two kinds of collisions in the patent DNA gene thing. One is business processes are much harder to patent, and natural things are much harder to patent. And they really collide with DNA sequencing and the process of using the sequences for disease determination. So right now, companies have a much harder time patenting the use of a particular DNA sequence for predicting a particular disease.
Right.
But believe me, many of the companies around here and elsewhere are trying to figure out ways around that, no doubt about it. And those things can change, right? They're only humans as judges that decide these things, right? They can change as climates change in intellectual property. Can you yourself patent your DNA? I'm not sure what the claims would be of that patent, right? What are you kind of saying the DNA does?
Produces someone awesome.
Yeah, of course. Not to be repeated, right? I mean, so remember, the point of patents remember, people think, oh yeah, it's greedy, I want to make money on a patent. The point of patents, though, is to disclose to the world what you know, so then they learn, right? That is the original point of a patent. So you don't keep it as a trade secret, which is the opposite, right? So I'm not sure how I would answer that, but there are companies still trying to patent genetic tests. Whether they're going to work or they're all going to get shut down remains to be seen.
Now this is interesting. What resources exist for tracking the latest developments in our understanding of genetics, and can we predict, for instance, if a malformed bone in a parent will be passed down to a child?
That's a great question. So a malformed bone, some DNA change associated with that, would it get passed on? Okay, so let's go through basic genetics too, because it actually makes a lot of sense. It's good to do this, right? So remember, every child has actually 6 billion letters, 3 billion from mom, 3 billion from dad, right? And our own 3 billion, I have 3 billion from my mom, 3 billion from my dad in those chromosomes. Chromosomes like chapters. All those pages go together. They're not really going to sort very far from each other. right? And so I have, for example, chromosome 1, chromosome 2, chromosome 3, all the way down. They're all mostly identical, except for the sex one. So XX for a woman, XY for a man. You get all that. So now, when I'm ready to have a child, I don't take my entire chromosome 1 and give it to my child. My chromosome 1 mixes up and then passes it on to the child, right? That's called recombination. So the child never gets the entire chromosome. They get a mix of your two, right? And it's always a different mix, right? That's every sperm and egg is a slightly different mix. So you cannot exactly tell, right? And things get even more complicated because remember, for any recipe, you got two recipes, one from the mom, one from the dad. You can't, just from the source code itself, if one of the recipes has a spelling problem, you can't really tell is there going to be a problem or not, right? So there are certain genes, for example, certain recipes that are only made from the father's version, certain only made from the mother's version. There's kind of chemical things that make that happen. So if there's a misspelling in the mother's version and it was not going to be used anyway, nothing happens, right? And it actually might get passed on, right? But nothing happened in you. So that makes genetics kind of interesting and fun and also super complicated. You can't just tell from the source code what is actually going to happen because it's like a procedure nobody ever calls, right? You could have a bug in there, but no one called it, right? So, if a tree falls in a forest, no one's there to hear it kind of thing, right? Does that make sense? That's a lot of DNA is like that. We just don't know. And then the worst part are, because we just haven't sequenced enough people on planet Earth yet, we got a lot of mutations or variants, same thing, where we just cannot tell what's going to happen, right? So we use all these bioinformatics algorithms and methods to predict, right? Because the recipes are cooking up a protein, and we think, okay, if it's spelled differently, the protein might change a shape here, it might become an acid, it used to be a base, it might be big, small. We try to predict how bad of a damage is it. It's basically a computer guess for a lot of these. And we use this phrase a lot called VUS, which is a variant of unclear significance. A lot of these are VUSs now because we just don't know. It's different. Maybe, we have 20 bioinformatics algorithms. Oh, how we wish they all gave the same answer, but they don't because different scientists have learned with different training sets. That is kind of the state of the art for DNA right now. We're way ahead measuring. We just don't have enough experience or measurements. We need those databases, right, to learn.
Yeah.
If we don't get those databases, we're never going to learn. What does that specific spelling difference mean for you?
Well, here's a question that maybe goes along with that, which is, does a person's genome change over time?
Yeah, does a person's genome change over time is a great question. So now we'll get to a little bit of kind of subtlety here, right? So epigenetics. So the DNA is the same DNA, but there are also chemical modifications of the DNA. So think of those like Post-it notes, like how I put a Post-it note on a flag of page. The cookbook might have some of those as well. And those could move. The book didn't change by moving the Post-it notes to figure out which recipes I like or not. Those can change as you get older. That certainly happens. Certainly those are tied to the chapters, so some of those get inherited. And here's what makes that worse. Those Post-it notes are different in different parts of your body. So there is no one epigenome. Okay, your blood will be different than the muscle, will be different than the bone. All of those are going to be different. So there's no one that you're going to test there. That makes it way harder to conceptualize what to do with this epigenome. But it is a flag of which recipes are getting cooked more often or less often. That changes over time too.
Yeah, I would imagine.
Yeah, and sometimes people say epigenome, but they mean environment. Right? Because scientists, it's still so hard to say environment if you're a biologist, right? It's so hard because then you got to figure out what people eat, how much exercise compared to something I could do in a petri dish.
You got to do all this messy stuff with like actual
ask people,
patients.
right? Or even look at the pictures of food. So it is hard for scientists sometimes to say environment, but it is clear it is environment, right? And so the environment plays a huge role.
So let's talk a little bit about some of these larger databases that are looking at doing these things. There some of them are coming from the government. You've got some of them coming from places like Google.
That's right.
Like what is the sort of high-level view of these long-term studies that might potentially help us to narrow some of this down? Yeah,
so I think a lot of research groups, health systems figured out DNA is where a lot of magic is going to be, and they wanted to be part of the solution of finding what these different differences might make. Groups, for example, giving full credit to a lot of my competitors and frenemies, as the kids would say, really friends at Vanderbilt, where they got community-wide approval to really get sequencing on. They have DNA on 500,000 patients. Mm-hmm. They're still running all the DNA. Children's Hospital of Philadelphia is a great example where they have lots of kids and parents I got a lot of DNA. Geisinger Health System, I'm on that advisory board. They're in the middle of nowhere, Pennsylvania, but they have a lot of folks that never move from that area. It's like they call it Iceland in the United States.
Are they the ones who work with the Amish communities?
That's right. Amish communities are there, but also others. The community really doesn't move, and they have 20 years of medical record data on those folks. They're trying to work with biotech companies to figure out this difference might mean this drug could work, things like that. And then you have newer groups, private organizations now. So Human Longevity in La Jolla, Aravale in Seattle. Those are early genome pioneers that launched those two companies. Everyone has, even Google with Verily has what's called a baseline study with Duke, Stanford, and Google trying to get a couple thousand individuals, not just DNA, but a lot of measurements. The thing to do is to find a population and really study to depth, to study in depth. To death, actually, literally. Yeah, actually, I didn't say the wrong thing. I said the right thing. Yeah, imagine that. There's a Freudian slip there. Now, the extreme of that is what President Obama talked about in the 2015 State of the Union address, and that is the next slide here, if you go back. All right, so State of the Union address, this has now been 4 years ago, where this is actually, so what happened in 2011 was a seminal moment where national academies were thinking about taxonomies of diseases, came up with this report. That's where this phrase precision medicine was used. And President Obama said we got to launch a precision medicine initiative. And kind of a random trivial fact, this isn't a White House thing for him. He actually proposed this for Illinois when he was a senator, actually, for the 1 year. So he's been thinking about this for a long time. This isn't just a buzzword that he was fed as a president. He was actually thinking about this. Now, you can see, the time is right, according to the slide, because we got abilities to sequence the genome, improved technologies for biomedical analysis, and new tools for using large data sets. And there's even a picture of a cloud there, believe it or not, right? This group knows 2 out of these 3, and you could argue 3 out of 3 are computers, right?
Hey.
This is why we're in this. And so part of this launched a lot of things. The National Cancer Institute should start something called NCI Match to get genome sequencing done on more patients matched on the clinical trials. The FDA should get extra money to figure out how to get precision medicine drugs approved. And we, as a country, should launch a massive 1 million or more volunteer study to actually try to get volunteers to contribute samples, including DNA, their medical records. And we promise to keep this in a secure, private, respectful way, but let's see what happens. How do you get diseases over 10 or 20 years? What exactly is changing so we can use that someday to prevent? And that's the next slide. They go back to this one. It's called the All of Us Research Program. This actually just launched 2 weeks ago. Some of you might have seen the advertising. If we were any good with the advertising, you might have seen some of it. I'm hoping some of you saw this. You can either go to an institution, University of California as a recruitment site, or you can just go to the joinallofus.org, and they'll shepherd you to your local Walgreens to be part of this as well. So pharmacies are involved, Google and Verily are involved with the app side to ask you questions about your health. And the idea is to get to 1 million people, and not to be too obvious about it, not 1 million people that all look the same, 1 million people that all look like us, like all of us, right? That's why it's called All of Us. And so to get to what we call underrepresented in research, URR communities that just never sign up for these things. And we got to get better to address all Americans. So that's what this is really all about. Now, here are the coolest things about this. The DNA is safe, the samples are safe, they're going to be processed. There will be data that gets sliced and diced and de-identified. Believe me, people don't want to know who you are here. They want to keep you sticky, right, to use the Silicon Valley term, to keep you on the apps. But we don't need to know who you are. We just want to know a lot of data. But even some of that data will be sliced and given back even up to the high school student level. The whole idea is to empower citizen scientists. It doesn't just have to be scientists that use this scientific data. Right? The whole point is that we can train a new generation of people who might not be scientists, but got to learn about science, right? So imagine all the science fair projects in the next 2 to 3 years coming out of Silicon Valley using these data sets. That is exactly what I'm seeing out of this project here. So it's going to be all of our data, safe, respectful, but really, really generating science from a new community.
So this brings me to another audience question. How do we protect our DNA from misuse by the insurance industry? For example, denying coverage for being a potential carrier of a particular gene. That's illegal, right?
That's a great question. It's not illegal.
Ooh.
Okay? No, so insurance is not covered. So GINA, again, what's the preface I'm going to say? I'm not a lawyer here, right? So Genetic Non-Discrimination Act is really more on the employer side.
Okay.
Life insurance is not one of those that's covered here, okay? This is where it gets sticky. At the same time, if you know your lipids are abnormal, if you know you're a smoker, you answer those on those life insurance questionnaires too, right? So DNA could be one of those. Let me just tell you though, the potency of DNA to predict any of those things, compared to seeing a picture of you smoking on Facebook, right? You tell me where the risk is here, right? So look, it is important to be mindful of your DNA, but how many times are we going to burn every paper cup we use, in a reception downstairs? Your DNA is behind. Is it worth it for me to sequence? No, I mean, why would I care here, right? But imagine that we are so giving of data that might show that we're obese, that we're smoking, us at a party drinking, and in some ways, it is absolutely legal to use that for risk determination from insurance companies. Yeah, DNA is there too, but I can tell you, we're not even close to reaching that same level of potency for disease prediction from DNA. We're just not. By the time you're an adult, you probably didn't get any of the childhood stuff that DNA is more potent for, right? Things like PKU, people have heard about the newborn tests. Those are all pretty potent tests. If you have that mutation, you're going to get that disease. A lot of those are the early ones. By the time you're an adult, you probably didn't have any of those. By definition, you're an adult, right? So the future prediction is a lot more about your environment. We're so sharing of pictures showing us in our natural environment today compared to the DNA. That's a long answer.
Well, speaking of our environment, we actually are also an environment.
Yes.
And so obviously there's a question here about how the microbiome
Perfect.
kind of fits into all of this.
Yeah.
Both into precision medicine and into our interest in DNA.
Absolutely. Okay, so microbiome are the passenger organisms around us, right? So we have bacteria living on our skin.
Right
Need
now.
any proof of that? I know as much as you wash your hands, just streak your fingers on the right kind of petri dish with the right jello in there and it'll grow. People know that. We're also compared with other orifices and the DNA, the bugs on both sides of those. And we can measure those. Now, so again, full disclosure, I'm involved with another company on that side too. I think it's going to make a difference, but we're not sure where yet. And this is one where I think patients are going to learn faster than doctors and researchers are here. I think the people that use these tests are folks with inflammatory bowel disease, irritable bowel syndrome. These are tough diseases. You can go on serious medications. A lot of times you don't want to be on those medications. And people are trying to find patterns, like I see these bugs coming, maybe that means I'm going to get a flare of a particular condition. And I think people are going to learn those patterns themselves faster than we're going to learn them for a population. So that's an example of precision medicine thing where I think each patient or individual is going to learn precisely what it means for them faster than we're going to learn a pattern for everyone, right?
Yeah.
But those are tests. And then there's the therapeutic side of that too. Do we take, is yogurt enough or fecal transplants enough? Those are therapies we use in hospitals now. We're trying those for some of these severe conditions. C. diff toxicity is a particular nasty bug that can lead to diarrhea. It keeps older people out of nursing homes. So we want to try to fix some of those things sooner. So those are all being trialed. Some of those have made it to actual approved therapies. But for regular people, we just don't know yet. But people can measure.
Yeah.
And again, I love that there are consumer companies that will help you measure if you want to measure, right? If it's your body, I think you should be able to measure some of those things.
Do you see any risks for genetic-based technologies in precision medicine in perpetuating inequality?
Yeah, absolutely. So inequality is there. Let me make a really obvious point, a couple points here for sure. Inequality. The best prediction of medicine working for you makes not a world of difference if you can't afford the medicine. Let's just make a statement like that, right? What good is it if you can't actually afford the medicine that's there, right? We have to get to those issues, no doubt. And so is it going to worsen things? Things are pretty bad now, right, in the world of medicine. The healthcare lack of system, right, that we'd call. So I think we have to address those, and they're going to get addressed. A couple kind of key points. I kind of hinted at one earlier. I think precision medicine is going to involve a lot of courage moving forward. What do I mean by that? It is so nice and shiny for us to say, boy, we got your DNA, this DNA mutation says you can get this drug, and here's this drug that's going to help you. Are we really going to have courage to say, the DNA says this drug is not going to help you and we're not going to use this drug.
We already don't do that.
We already don't do that, right?
That's right.
And so what does that exactly mean? Because we're entering a world where precision medicine drugs are going to be more and more costly. You can argue that's needed. You can argue that's the thing about the discovery world or not, but they're certainly going to be more costly. What does it mean if we're going to use more costly drugs on patients that actually still have some hope, want to have some hope, but every test is saying that that drug is not going to work? That's what I mean by courage. I'm not sure who's going to show that courage, to be blunt right now, right? So unless we think that way, there might be more expenses here, right? So I think that can worsen disparities as well here. And then in the end, too, it's frustrating, right? That you got a cancer, let's say, look at Duchenne's muscular dystrophy. Let's get off cancer for a second, where we have a drug for Duchenne's muscular dystrophy for 4% of those patients, or cystic fibrosis is a better example. We have a drug for 4% of CF. Already CF is rare, and 4% of them can benefit from a drug, and maybe another couple percent based on their DNA. Boy, I mean, that's the most frustrating thing in the world. You got hit with this rare disease, and you see a few of these kids actually benefiting, adults benefiting, and what about me, right? What about everyone else? So it's disparities in a very different way, genetics, but in a very different way. So the kind of truism there, I mean, I'll say this, and I'm not saying this to be kind of evil or anything. We have drug companies and pharmaceutical companies. We don't have enough pharma and biotech companies on the planet right now, okay? To me, I'm not saying that because we need more greed and revenue. We need to get to drugs in a way more efficient way, right? And to me, that is a compute problem. It's a data problem. It's a coding problem. Can we get to ways to get to new drugs? Because there are just not enough pharma and biotech companies on the planet to get to every sub-disease we're going to be partitioning out in precision medicine, right? We've got to get to more efficient ways to do it. I would not be surprised if someone in this audience has a novel way to do it. There's open data out there, and we just got to get to more of those. Otherwise, there's just there are not going to be enough drugs for everyone with every one of these sub-diseases.
So you'll be in the position of I know what's wrong with me, but there's nothing I can do about it.
There's a lot of that, and that's where there is this fear from the medical industry that built-in anxiety and frustration, right? Why get a test that you can't do anything about? Now, look, I get it. A lot of different opinions there. We were involved with first family to get their genome sequenced, a Cupertino family, and this, again, not a usual family. The father ran a sequencing company, sold it to Illumina, John West and his family, and they're very public about this. And part of that was a 11th grader at the time. And she was in the Wall Street Journal. She actually presented her genome at Cold Spring Harbor, where all these scientific findings were done, as an 11th grader. And someone from Wall Street Journal asked her, aren't you afraid you're going to learn about something that's going to be lethal to you in 20 years? Why aren't you afraid of that in your DNA? And you know what she said? This was already 10 years ago. She said, if I knew I'm going to get a disease, I'm going to start today having bake sales. I'm going to raise some money to get some research in this disease, right? And kids are not that fatalistic, right? They have no doubt that someone smart, given enough resources, can fix this thing in 20 years. And so sometimes we should think like the kids do.
Okay, so how would you structure clinical trials for these kinds of rare diseases where the incidence is so small that it can be difficult to draw statistically significant conclusions?
Yeah, rare diseases are going to be tough, right? And so just to make an obvious point, so rare disease, I think there's an official definition of less than 1 in 50,000, but in that number, there are still tens of thousands of rare diseases in the United States alone, all with their little codes or lack of codes, right? The trials for those are going to be tough. And so one of the things I'm really pleased about, one way I think we're going to make a difference is by getting medical centers that see these kinds of patients to work together. And why I love, and I'll put in a plug for University of California for a second, why I love being at UCSF is in some weird way we're a test bed for health systems working together, right? So UCSF is one of 6 medical schools in the University of California, right? UC is 10 campuses. 6 of them are medical, 4 are not medical. So Santa Cruz, Berkeley, they're not medical. The other 6, UCSF, UCLA, Irvine, Davis, San Diego, Riverside. I've memorized that list. And we actually have something called UC Health where we all act together. So for example, if you're a pharmaceutical company working with a doctor to run a trial, you get IRB approval to go ahead in one of the 6, you'll get automatic approval in the other 5, right? I mean, we can scale, right? The hardest thing to do in Silicon Valley is scale. We help folks scale in this way. I think that's going to be the future with these health systems working together to get those rare folks into meaningful numbers so we can actually run a trial. Now, there are going to be some conditions that are just N of 1, right? What we say, there's no one else that we know of in the country that we can run a trial on, or N of 2. And a lot of those are going to be parents and patients and support groups organizing those communities. And some are going to be apps and social communities and even good old Facebook to help those folks organize each other. But I think the future, there's a huge future in parents and patients kind of understanding much more. The one story I will tell you, and it's really kind of amazing, heartwarming story. We had one child here, they're very open about this. It was on CNN, the kid who would not cry. And a couple years old, trying to figure out what's going on with this child, having liver abnormalities and muscle weakness, not cognitively developing, got a genome sequence, one of these early kids. We figured out it could be these 50 things, right? And what did we do differently here? We gave the list to the dad and mom, right? And what do? They start to Google some of these genes, and boy, they find another blog post from another parent who just got the same list over on the East Coast. And way faster than us as scientists writing papers, they found each other, and now there's 2, right? And now there's 20, and I don't know how many are in NGLI1, right? That's what it means to give data back to parents and patients, controlled ways, with the right education, all that, respectful way to do it. But it's not about holding on to all this data, because nobody cares more about being sick than someone who's sick, right?
Well, I mean, this leads me to the next audience question, which is how the digital age might help us to manage our own minor medical issues without ever having to go to a doctor.
Yeah, absolutely. So, it's crazy, and I'll put in a little plug for myself here for a second, right? So I used to laugh at quantified sulfurs. I'm sure there are a bunch of people in this audience. I saw some Apple Watches, that's one extreme. There are Fitbitters and all the rest, quantified sulfurs. Their early meetings were here, I think, right? So I used to laugh at these folks. Measuring everything comes in and out of your body, right? So measuring things that go in sounds tractable. Measuring things that come out sounds kind of gross to me, but but it's doable, right? So I used to laugh at them until December of 2012. Okay, came back from a trip to Hawaii, and I stepped on a scale, and it was 247 pounds. And then I realized when I hit 250, they can measure me in tons, right? That is 1/8 of a metric ton. I was like, what the hell happened to me, right? I used to walk all over in Boston. I'm driving out everywhere in Silicon Valley, right? So again, not to put in plugs, I'm not financially connected. I bought everything Fitbit makes, including the scales, and I lost 50 pounds with those things, right? And I have a whole TED thing on this, right? So I'm a believer in this now, and I record everything I eat. So that little sandwich I had in there, I got to write down at some point. I've weighed myself every morning for 5 years. I've written everything down. I've kept most of the weight off. So I'm a believer that you can be empowered by using your own data. Now, look, I'm guilty of making everything seem easy. I admit that, right? Losing weight is the hardest thing in the world. Quitting smoking is the hardest thing in the world. But I'm a firm believer that digital devices will help you do it. But if it's not Fitbit, it's something else. Now, here's the funny part. Everyone on Facebook knows I lost 50 pounds. I'm not shy, if you couldn't tell, right? But my doctor, right, whose medical system, I'm not going to say which practice they're in, spent literally $1 billion on Epic, which is their EHR system, was the last one to know, because their $1 billion system doesn't talk to my $40 gadget, right? Can you just smell the opportunity here, people, right? And we in medicine know we have got to get with the program, because if we don't stay on top of these devices and the data behind them, we're going to lose healthy people to Silicon Valley, right? I mean, there are other ways to take care of them. Now, at the same time, doctors don't want to be inundated with more data. That setup isn't there. So we're going to need digital tools on our end to help manage populations with these kinds of devices. And you can imagine there are no companies really in any of this space yet. It's all green field right now. What exactly do you deliver to a doctor? How much do you manage by yourself as an algorithm before bunting up to the doctor? All of those things we're still trying to figure out.
Well, this brings me to our last question from the audience, and it is, as a community-based oncologist, how do I prepare for the future of precision medicine today?
Wow, community-based oncologist. So I don't envy oncologists in particular right now. I've been to some of the continuing medical education classes. It is rough right now. So for example, we have immunotherapies, right? And there's combinations of immunotherapies. And even just a basic probability in combinatorics, n and n squared and all sorts of ways to pair these things. I think there are 1,000 immuno-oncology trials underway in the United States, according to the New York Times a couple weeks ago, right? And just keeping track of which patients qualify which trials seems incredibly rough to me right now. I think that community in particular is going to be depending on digital devices even sooner. So these are going to be decision support tools that insert themselves. There are probably like a dozen startups plus or minus 10 miles from here that are working on digital tools that try to give advice through Epic or Cerner, the electronic health record systems. Some of that advice will come when you send the genome to a company. They're going to send you back trials that a patient might, literally on the fax. And yes, we still use faxes in medicine. You all know that. DNA by fax, right? Think how crazy that is, right? But that's how we get it because it works, right? You don't have to fiddle with projectors and stuff. So, I mean, I think that's where most of this advice is going to come through computers, through companies. But it is going to be impossible to keep everything in your head that much longer.
So, and I'm just imagining I'm a doctor. I like to pretend to be other people sometimes. But like, would it make sense to start asking your patients if they're doing, the sort of quantified self, any kind of data tracking, those sorts of things, and maybe begin having those conversations with them?
Yeah, so I think in some ways, the data tracking is going to really make a bigger difference for primary care physicians. I think, so certainly there are going to be aspects if you have cancer, make sure that your weight is okay, things like that. Things like that, I think, so for example, you can track your parents, right, with Fitbits and things like that. Not to be invasive, but just to make sure they're walking okay, stuff like that. These are really low-cost medical devices in some ways. So I think tracking, especially for primary care and wearables, are going to make a difference if we're ready to accept that. Now, that being said, there have been studies run, controlled studies showing patients that get these wearables do not lose weight. Actually, they gain weight, okay? At the same time, so think about why is that? At the same time, I'm thinking, how much counseling and advice does the patient get, or do they just get handed a Fitbit box, right, and go try this, right? So I don't think we're even ready in medicine to know how to even give these devices and counseling and getting people started on this, right? It's like plugging in a VCR, getting your Wi-Fi working. Some expert knows, the rest you don't know, right? So.
Well, it's not just that, though. It's also the actual design of the devices. They're not necessarily as sticky as you might like. Sometimes they just feel like punishment. And
absolutely. So especially if you don't like the numbers it's giving you.
Yeah.
Right? So. But there's still a lot of low-hanging fruit out there, okay? So for example, I've started to look at the patterns for myself. Like, for example, again, not to put in plugs too much, but there's this one restaurant on University Ave in Palo called, it's on Hamilton Ave called Reposado. It's a high-end Mexican restaurant, right?
It's delicious.
It's delicious, and the guacamole is to die for. But I know every time I go to Reposado, I know I gain weight the next morning. Okay, like I've done it again and again and again. How many more? And those chips are just amazing, right? So I've learned a pattern. Yet there is no app still that just tells me as I'm GPS walking into Reposado, can you try to order something else? Right? Every bit of the tech is there in the phone, but that app doesn't exist still, right? Someone's digitized the menu somewhere. Someone knows the calorie counts for everything, and still that app, and many others still haven't been written yet, right? So I think we have a glorious future, but learning about where the need is still the hard part, not writing the code, right? I mean, figuring that out. And look, I'm giving away answers and ideas here. Go, if you did that, I'll pay 99 cents for that, right? I mean, not much more though, but.
All right, well, thank you so much.
Thanks, thank you all for tolerating us.